the most complex part of the human body. With over 100 billion neurons, this 3-pound organ is the seat of intelligence, interpreter of the senses, initiator of body movement, and controller of behavior. Lying in its bony shell and washed by protective fluid, the brain is the source of all the qualities that define humanity.
For as long as I can remember, I've always been fascinated with the human brain. Hi, my name is Fida, and I met Mark while I was doing my thesis for my PhD, and I discovered that we both share the same passion for discovering the secrets of the human body's final frontier.
So let's start off with a history tidbit. You see, approximately 130 years ago, a scientist named Paul Ehrlich inserted a dye into the bloodstream of a mouse and made a groundbreaking discovery. He discovered that the dye spread through all tissues except the spinal cord and the brain. He realized that there was a barrier around the brain that allowed specific molecules such as oxygen, glucose, and amino acids to travel through while protecting the brain from neurotoxic compounds and pathogens. The man who discovered this blood-brain barrier was actually my grandfather, and he inspired me to continue on his work.
Let's go deeper into the blood-brain barrier. In fact, how good is this barrier? To give a more detailed definition, the blood-brain barrier is an important mechanism of the central nervous system that filters out certain substances inside blood to prevent them from harming the brain. It selectively allows individual molecules, such as small lipid-soluble molecules, from blood to enter the brain, and vice versa. The blood-brain barrier is formed from tightly packed endothelial cells of the capillary wall, which prevent the passage of solutes, while astrocytes provide biochemical support to these cells.
But why is this important? Why are we talking about the blood-brain barrier if this perfect system is already in place? The reason is neural diseases. In order to be able to treat diseases such as Alzheimer's or Parkinson's or Huntington's, we have to develop drugs that can go through the blood-brain barrier without causing harm to it. However, this is where another problem presents itself. How can we test these drugs?
In order to develop drugs that can cure neural diseases without causing damage to the blood-brain barrier, which naturally defends against invaders, the drugs need to be tested in an environment that fully represents the blood-brain barrier in vivo. Human brains cannot be tested on, and animal testing raises an unethical and moral issue. So what is there to do?
This is where biomimicry comes in. Biomimicry, like the name suggests, is the design and production of materials, structures, and systems that are modeled on biological entities and processes. In other words, you model something based on nature. So, if you can artificially recreate the blood-brain barrier, you can repeatedly perform experiments, drastically shortening the time it takes to test drugs, components, and nanovectors, all without the use of live animals. Prior to the existence of all this modern technology, trying to artificially recreate the blood-brain barrier was virtually impossible. But Feed and I have accomplished something brand new for mankind, and we're here today to share the information with you.
We have successfully engineered high-resolution 3D printing that is able to reproduce microcapillaries of the human neurovascular system on a 1-to-1 scale. Here is a scanning electron microscopy image of our blood-brain barrier model. And as you can see, the artificial structure consists of 3D porous tubes with a diameter size of typical brain microcapillaries, which is about 10 micrometers. Microtubes inspired by brain capillaries were fabricated through 2-photon lithography, a process in which a liquid resin hardens when it's exposed to laser light, making the structure very precise.
The microtubes, as you can see over here, are then used as scaffolds for the co-culturing of endothelial-like BN3 and U87 glioblastoma cells in order to simulate the blood-brain barrier. In order to further mimic the in vivo conditions, the flow rate of the solution inside the luminal microvessels is set to a value comparable to that of brain microcapillaries. Not only that, but the in vitro model provides a closed system where the different variables such as drug concentration, blood flow speed, pH, and temperature can be easily tuned and monitored, thus providing precious and detailed information about the blood-brain barrier crossing in real time and at a cellular and subcellular level. Thanks to biomimicry, we were able to develop a biohybrid microfluidic model of blood-brain barrier to allow for testing efficiently and ethically.
Sure, we can now mimic the blood-brain barrier, but what are some applications? Well, this is where nanotechnology comes into play. I know, it sounds like something from the future, but really, it is just technology that deals with structures of small size, specifically structures less than 100 nanometers.
You see, the main application of our modeled blood-brain barrier would be nanomedicine. Since it is unethical to test on living organisms, scientists can use this model to test drugs that target neurological diseases. Since the vast majority of potential drug treatments do not readily cross the blood-brain barrier, they can pose a huge impediment to treating mental and neurological disorders.
Now, researchers can use our device to understand the interaction of drugs or of drug delivery nanovectors to overcome the blood-brain barrier and target the central nervous system. Now, don't get me wrong, the use of nanomedicine is not new. In fact, Chinese scientists at the Shanghai Institute of Materia Medica have conducted a study where they used albumin-binding proteins to explore the blood-brain barrier.
In their study, scientists managed to identify a potential cure for brain cancer. Scientists found that by attaching drugs to albumin, the drugs could bypass the blood-brain barrier. Albumin is actually a protein made by your liver that helps keep your fluids in your bloodstream so they don't leak into other tissues, and proteins such as Spark and GB60 actually bind to the albumin and carry the albumin as an amino acid. The catch is that this amino acid is also an energy source for many types of brain cancer tumors. If drugs are attached to the albumin will lead to the drugs to brain cancer tumors, which will be able to destroy the tumors at their core. This shows that that such pathways could be explored for the use in brain-targeting biomimetic delivery of efficient cancer treatment.
This study is a major breakthrough in brain cancer treatment. So why haven't we heard about it yet? The reason is because this study was carried out on mice. Clearly, mice are not the same as us human beings because they have differences in their anatomy, thus making the nanomedicine solution much less pleasing to carry out on humans. I mean, which rational human being would use drugs tested on mice? With our biomimetic model, the blood-brain barrier, such experiments can be carried out without the potential harm of living organisms. Moreover, they're tested in a full environment that equally resembles the real blood-brain barrier. Think about the possibilities of developing drugs and testing drugs that bind to proteins to cross the blood-brain barrier easily and treat neurological disorders.
So we'll leave you with some things to think about. Recreating the human brain and curing neurological diseases without side effects is a big task, but it's just the tip of the iceberg. Imagine the whole plethora of applications with nanotechnology and biomimicry that have yet to be unveiled. With a little bit more time and resources, we can see great innovation that can help everyone. Thank you.